I'm Dr. Rebecca James, author of the book Self-Assessment and Review of Biochemistry by JP Publications. Today I am here to discuss some questions that came for the last 2 years, PGME, PGI, AIIMS, etc. in the topic of amino acids. So we'll straight away move on to the review questions. The first question, amino acid with hydroxyl group. Options are threonine, tyrosine, serine, tryptophan and valine. So to answer this question, we'll see the classification of amino acids based on its side chain. Based on the side chain, amino acids are classified into two, the aliphatic amino acids and the aromatic amino acids. Among the aliphatic amino acids, the number one is the simple amino acids. They are glycine and alanine. Then the branched chain amino acids, that is leucine, isoleucine and valine. Then comes the sulfur containing amino acids, that is cysteine and methionine. Then comes the hydroxyl group containing amino acids. The hydroxyl group containing amino acids are serine and threonine. Then in the aliphatic amino acids, the other groups are the amide group containing amino acids that include glutamine and asparagine. Then the acidic amino acids, which include the aspartic acid and glutamic acid, and the basic amino acids, that is arginine and lysine. So now we move on to the second group of the amino acids, that is the aromatic amino acids. Aromatic amino acids include number one, the phenylalanine, which contains the benzene ring. And this benzene ring is hydroxylated. The product is tyrosine. So the tyrosine is having a ring structure with the hydroxyl group, that is the phenol group. This phenol group is having hydroxyl group. So hence tyrosine is also a aromatic amino acid with hydroxyl group containing with hydroxyl group, and then the histidine and tryptophan. So that along with it we have to learn one amino acid which we cannot classify under aliphatic or aromatic, and that is the proline. The proline contains the amino acid, and the ring structure present in proline is a pyrrolidine ring. So now we see that question again. Amino acid with the hydroxyl group, they are threonine, tyrosine and serine. Next question is, the fibropeptidase A and B are highly negative due to the presence of which amino acids? It's a recent year question to January. The options are glutamate and aspartate, serine and threonine, lysine and arginine, valine and lysine. So among this option, we have to search an amino acid with negative charge. So which are the negatively charged amino acids? The negatively charged amino acids are the aspartic acid and the glutamic acid. The aspartic acid is otherwise called as aspartate and glutamic acid is otherwise called as glutamate. So going back to the question, fibropeptidase A and B are highly negative due to the presence of glutamate and aspartate, which are the negatively charged acidic amino acids in the list. Next question is, which of the following amino acid is not formed by post-translational modification? So first we should know which are the amino acids that are formed by post-translational modification. The amino acids that are formed by post-translational modification are the derived amino acids. So option A, triiodothyronine, is actually an amino acid. It is thyroxine, which is the tyrosine group in the thyroglobulin is iodinated and it is forming the triiodothyronine. So it is not amino acid with no post-translational modification. Hydroxyproline and hydroxylysine, definitely they are the derived amino acids and they are formed by post-translational modification. And what is remaining now is the option D, that is selenocysteine. So we'll see how selenocysteine is formed. The selenocysteine is the 21st amino acid. It is seen in the active site of thioredoxin reductase, glutathione peroxidase, deiodinase, selenoprotein P. Now we'll see how this selenocysteine is formed. Selenocysteine is formed, or it is coded by a stop codon, that is UGA. How a stop codon can code for an amino acid? This is by a process which is called as recoding. What is this recoding? In the mRNA, where it has a UGA, this UGA has to be converted to selenocysteine. Then in those mRNA, there is an element in the untranslated region, and that is called as the Sekis element. So if the Sekis element is present in the 3' untranslated region, and in that mRNA you can see here a UGA codon. UGA is actually a stop codon, but in this particular case the UGA is it is bind with a tRNA. And what is the tRNA that binding that to this UGA? The tRNA that binds to this UGA codon is a tRNA which carries serine. Now how the serine is converted to selenocysteine? The serine, as and when the translation is occurring, that means co-translationally serine is modified to selenocysteine. So the selenocysteine is an amino acid which is formed by co-translational modification and not a post-translational modification. So among the option, now we are going again to that question, the special amino acid not formed by post-translational modification is selenocysteine. Now another question that is about the selenocysteine is selenocysteine is coded by. Selenocysteine is, we have seen it, it is coded by the stop codon UGA. Now a question in the general amino acid metabolism. It is phenyl butyrate is used in the urea cycle disorder because. The answer here is, the options are scavenges nitrogen, increases enzyme activity, maintain energy level, increases renal output of ammonia. The answer here is it scavenges nitrogen. We'll see how a phenyl butyrate scavenges nitrogen. This phenyl butyrate is a prodrug. So as in the body, this phenyl butyrate is metabolized to phenyl acetate. And this phenyl acetate combines with glutamine. What is the peculiarity of glutamine? Glutamine is the carrier of amino groups or ammonia or transport form of ammonia in our body. So this glutamine is in fact rich in the amino groups or the nitrogen. So once the phenyl acetate combines with the glutamine, it forms phenyl acetyl glutamine and it is excreted through urine. So the answer for that question, phenyl butyrate is used in the phenyl butyrate is used because it scavenges nitrogen. Serotonin is, options are 5-hydroxytryptophan, 5-hydroxytryptamine, 5-carboxytryptamine and 5-carboxytryptophan. So we should know the chemical name of serotonin, then only we can answer this question. And the answer is 5-hydroxytryptamine. So we'll see how this serotonin is formed. The tryptophan is first hydroxylated to 5-hydroxytryptophan. This is by the enzyme tryptophan hydroxylase. Then the 5-hydroxytryptophan is decarboxylated to 5-hydroxytryptamine. And this 5-hydroxytryptamine is nothing but the serotonin. So the question was serotonin is 5-hydroxytryptamine. Now another question that came for recent year January 2018 is Tyros Enosis, the most common causes. Fumarate acetoacetate hydrolase, tyrosine transaminase, para-hydroxyphenyl pyruvate hydroxylase or homogentisate oxidase. So the answer here is Tyros Enosis, the most common type is Tyros Enosis type 1, which is otherwise Tyros anemia type 1, and it is caused due to a defect in fumarate acetoacetate hydrolase. So for that we'll see the pathway of phenylalanine catabolism. The phenylalanine is initially hydroxylated to tyrosine. The enzyme is phenylalanine hydroxylase, and this phenylalanine, which is hydroxylated, that forms tyrosine, it is transaminated to para-hydroxyphenyl pyruvate. Then the para-hydroxyphenyl pyruvate is converted to homogentisic acid. The enzyme here is PHPP hydroxylase. Then the homogentisic acid is converted to mallyl acetoacetate by the enzyme homogentisate oxidase, and the mallyl acetoacetate is isomerized to fumaryl acetoacetate, which is later on hydrolytically cleaved to fumarate and acetoacetate by the enzyme fumaryl acetoacetate hydrolase. So here in this pathway we will see what are the causes of Tyros Enosis. There are three types of Tyros anemia or Tyros Enosis. The Tyros Enosis type 1, we will take it as the first Tyros Enosis or first Tyros anemia. We know that the first, it's starting with the letter F. So in this pathway, the Tyros Enosis type 1, you can remember, it is an enzyme deficiency that starts with the letter F, and it is fumaryl acetoacetate hydrolase. Next is the Tyros anemia type 2. The Tyros anemia type 2, remember T and T is there, and the Tyros anemia type 2 is due to a defect in the enzyme tyrosine transaminase. This is type 2. Then comes the Tyros anemia type 3. So to find out the enzyme deficiency in the Tyros anemia type 3, we will find out an enzyme with the 3 P's in its name. So that is PHPP hydroxylase. So if this is defective, it causes type 3 Tyros anemia. And among the type 1, type 2 and type 3, the most common one is type 1, hence the answer is fumaryl acetoacetate hydrolase. Now another question is 5-HIAA is excreted in urine due to. This is also a recent year 2018 January question, and the answer here is Carcinoid Syndrome. So a few words about Carcinoid Syndrome. Carcinoid Syndrome is due to a defect in the argentaffin cells, that is, there is a tumor of the argentaffin cells. What is the function of the argentaffin cells? The argentaffin cells are the cells that secretes or synthesizes serotonin. So if there is a tumor of the argentaffin cells, there is increased synthesis of serotonin, and that leads to increased serotonin, and the serotonin, the excreted product is 5-HIAA, that is 5-hydroxyindole acetic acid. So if there is increased serotonin, that leads to increased 5-hydroxyindole acetic acid in the urine. So hence the answer is 5-HIAA. Now the next question which came for the January 2018 exam is PKU, the true statement is. Actually this is a poor recall, the options I couldn't get completely, but still we will discuss a few words about the PKU. The option 1 was neurological symptoms are due to excess phenylalanine in the blood. Blood phenylalanine more than 20 mg per deciliter is suggesting bad prognosis. Urinary phenylalanine is used for the screening. So here we will see the first option, that is the neurological symptoms are due to excess phenylalanine in the blood. What is the cause of the neurological symptoms in PKU? The neurological symptoms in the PKU is basically because of the excess phenylalanine in the blood, and this phenylalanine is a bulky amino acid. This bulky amino acid competitively inhibit the entry of other bulky amino acids like tyrosine, tryptophan through the blood-brain barrier into the brain. So this causes a deficiency in the neurotransmitters that are formed from the tyrosine and the tryptophan. That is one of the cause for the neurological symptoms in PKU. But here the question is neurological symptoms, and in option here it is neurological symptoms are due to excess phenylalanine in the blood. It is not directly due to excess phenylalanine in the blood, but the excess phenylalanine which is inhibiting the entry of bulky amino acid into the brain, that is causing the neurological symptoms. So we cannot take that as the answer. The next option is clearly given in the Nelson as the phenylalanine more than 20 milligram per deciliter is considered as a bad prognostic sign. And the last option, urinary phenylalanine, is used for screening. Actually, for the screening of PKU, the sample that we take is a blood sample, not urine sample. So here the best possible or the true statement here is the option 2, that is blood phenylalanine more than 20 milligram per deciliter is suggesting a bad prognosis. So here we will discuss some of the biochemical defect in the phenylketonuria. So for that we learn the pathway of how the phenylalanine is converted to tyrosine. The enzyme here is phenylalanine hydroxylase. So when this is happening, what happens on the other side is tetrahydrobiopterin is converted to dihydrobiopterin. The enzyme here is dihydrobiopterin reductase. Dihydrobiopterin reductase. And this dihydrobiopterin has to be converted back to tetrahydrobiopterin, and for that we need the enzyme dihydrobiopterin reductase, and this require NADPH. For this it require NADPH. So what are the biochemical defects that can cause PKU? One, the defect in phenylalanine hydroxylase defect, it causes the classic PKU. Then the non-classic PKUs, there are so many causes, but the main important cause you can say is the dihydrobiopterin reductase deficiency, which is causing a defect in this cofactor. So the classic PKU is due to a defect in the phenylalanine hydroxylase, and the non-classic PKU is due to a defect in the dihydrobiopterin reductase. And in other words, the non-classic PKUs are due to a defect in dihydrobiopterin reductase, or you can say it is due to a defect in the synthesis of this cofactor, that is tetrahydrobiopterin. So we'll see a question that is that hyperphenylalanemia occurs due to. This is hyperphenylalanemia can occur due to option A, that is phenylalanine hydroxylase deficiency. Yes, that is causing the classic PKU. Then the next option is phenylalanine hydroxylase overactivity. No, it is not overactivity, it is deficiency. Option 3 is dihydrobiopterin reductase deficiency. Definitely a deficiency of dihydrobiopterin reductase causes non-classic PKU. And option 4 you can see tyrosine hydroxylase deficiency. It will not cause hyperphenylalanemia. And final option is defect in dihydrobiopterin biosynthesis. And the cofactor biosynthesis is affected in non-classic PKU. So the answers are phenylalanine hydroxylase deficiency, dihydrobiopterin reductase deficiency, and a defect in dihydrobiopterin biosynthesis. Now another question in the sulfur containing amino acid is, which is the amino acid not excreted in cystinuria? Options are lysine, ornithine, cysteine, and cysteine. So here we will see something about the cystinuria. In the case of cystinuria, the biochemical defect is a defect in a dibasic amino acid transporter, and it causes excretion of cola in urine. So what is this cola? Cola is cysteine, ornithine, lysine, and arginine. Remember, in cystinuria, what is excreted is cysteine and not cysteine. And apart from that, you can see cyanide nitroprusside test is positive, and common disorder associated with amino acid malabsorption, And one more thing is, this is an amino acid urea you find in the Garrod's tetrad. So in the question, which amino acid is not excreted in cystinuria is cysteine. Cysteine is never excreted, but what is excreted is cysteine. Another question that came for the January 2018 exam was, fish odor syndrome can be prevented by the intake of? The answer is choline. So what is this fish odor syndrome? The fish odor syndrome is a disorder due to a deficiency of the enzyme called as trimethylamine oxidase. This is a flavin containing monooxygenase, and what happens here is, because of the deficiency of this enzyme, the trimethylamine excess or it is accumulating in our body. This causes a foul smell of rotten fish. How can we prevent this disorder? This can be prevented by the intake of choline-rich food, which include fish, egg, liver and other sources of choline. So coming back to the question, trimethylamine or fish odor syndrome, what should be avoided from the diet is the choline. So today we have discussed some questions that came for the recent PGME in the amino acid metabolism. So one thing that I want to tell you here is, amino acid metabolism is the most repeatedly asked question for various PGME exams. So never forget to learn the amino acid metabolisms, and if you have any doubts, you can always contact me. I am always available for you in the FB Messenger, and the amino acid metabolisms is very frequently asked. One more reminder I want to give you, and I'll come back to you with more videos on carbohydrates, lipids, etc. Thank you.